Hello and welcome to the channel. We want to continue our study on antimicrobials, specifically on antibiotics. So we ended at looking at the mechanisms by which antibiotics work. Generally, we said antibiotics can be cidal or static, bactericidal or bacteriostatic. In other words, antibiotics can kill bacteria or stop the growth of bacteria. Now, I want to look at the general mechanism by which these antibiotics are able to achieve the results. There are primarily three ways by which antibiotics are able to achieve the results. There are several other ways which can fit in here, but we want to look at the general. There are about three ways. The first, most prominent, is antibiotics work by preventing cell wall synthesis, preventing or inhibiting cell wall synthesis. In other words, prevent the bacteria from making cell wall. Now, if you still remember, we said bacteria is a single-celled organism. It multiplies or reproduces by dividing itself. So if it divides, it must now have two new cell walls to accommodate the two new baby bacteria, if you like, daughter bacteria. So in other words, each time bacteria must grow or reproduce, it needs a new cell wall. Now, to synthesize this new cell wall for any breakage, for any replication or reproduction activity, bacteria goes through a process. And our class of drugs we are describing here, antibiotics, some class of antibiotics, works by preventing or inhibiting the making of the bacterial cell wall. Example of such includes the penicillins, the cephalosporins, the monobactams, the glycopeptides, etc. Now, another way some antibiotics work is by inhibiting enzymes that bacteria requires for its activity. So two main activities we interested in. One is protein synthesis. Bacteria must synthesize proteins in order to grow, multiply. And so it uses enzymes, which are known as biological catalysts, so that it goes through reactions to create the proteins. We have antibiotics that are able to interfere with the protein synthesis. Another group of antibiotics also interfere with DNA synthesis. Each time there is a reproduction, there must be a copy of the parent DNA. Enzymes are required for the DNA copying or replication. And antibiotics, there are some antibiotics that are available to interfere with this replication process. So that means there are three ways, primarily, generally, by which antibiotics work. So if we put those three ways into a picture form, what we are saying is we have those that are cell wall synthesis inhibitors, which means these deal with the peptidoglycan, main component of the cell wall, which is in both gram-positive, gram-negative organisms. So a class of antibiotics that deal with cell wall synthesis inhibition includes the beta-lactams, And there are four main members of the beta-lactams, so penicillins, cephalosporins, carbapenems, monobactams. They are all cell wall synthesis inhibitors. Then we have the glycopeptides, vancomycin. We have bacitracin. Now, these are also cell wall synthesis inhibitors. Then we have the protein synthesis inhibitors. Bacteria uses this structure here, which is called the ribosome, for its protein synthesis. Now, there are two main components of the ribosome, the 50S ribosome, the 30S ribosome. Each subunit works hand in hand with the other to make protein synthesis. So the two must all be functional to create your protein. Now, sometimes we have antibiotics that interfere with the 50S activity and hence leads to faulty protein synthesis, or interferes with the 30S ribosome subunit and thus interferes with protein synthesis. There are two main antibiotics that work on the 30S, tetracyclines and aminoglycosides. And then for those that work on 50S, macrolates, lincosamides, chloramphenicol, those are the ones we will focus on. And the final group is the nucleic acid synthesis inhibitors. Now, as the name indicates, these are into nucleic acid synthesis. And when we say nucleic acid, basically DNA. So we have two. We have those that are direct DNA synthesis inhibitors. Now, this is the DNA of the bacteria. So in that case, the quinolines. And we have various, we have two main enzymes that are inhibited here. We will go into that detail later. Then we have the indirect DNA synthesis. Now, in the case of indirect DNA synthesis, notice that here in the picture, this enzyme inhibition is actually direct on the DNA. But in the folic or the folate synthesis inhibition, notice that it is indirect because this inhibition of this process here prevents the folic acid needed to be inculcated into the DNA. And therefore, without the tetrahydrofolate, DNA that is formed is faulty. So it's indirect, not direct on the DNA, indirect, but both instances, folate and direct DNA synthesis inhibitors, are all nucleic acid synthesis inhibitors. So now these are the general ways by which our antibiotics work. And if we put that into the chemical structure, then we can see that for cell wall synthesis inhibitors, we have the beta-lactam antibiotics such as penicillins, cephalosporins, carbapenems, monobactams, and glycopeptides. Note they all inhibit cell wall synthesis. Then we have the protein synthesis inhibitors, and these inhibit protein. Remember, they can do it either at the 30S or the 50S subunit. And there we can use MALT to help us remember. Macrolates, aminoglycosides, lincosamides, and tetracyclines. So I like to say MALT gives protein. MALT gives protein. Basically, the idea is that inhibiting MALT inhibit protein synthesis. And then we have the nucleic acid synthesis inhibitors. We have the indirect sulfonamides and the direct fluoroquinolines. Note here that fluoroquinolines are the only direct DNA synthesis inhibitors. They hit DNA direct through specific enzymes, and we will go into those enzymes as well. So let's just look at these three mechanisms. Let's look at maybe one or two and see how it works. So now let's take this general picture again. Now, this is the cell wall of gram-positive bacteria, gram-negative bacteria. Now, again, we notice that there's the peptidoglycan in both gram-positive and gram-negative. Howbeit in gram-positive, it's thicker. In gram-negative, it is narrower. Now, when you look at the peptidoglycan, that is what is known as the cell wall. So in gram-positive, cell wall is made primarily of peptidoglycan. Then in gram-negative, the cell wall is made up of a combination of the peptidoglycan and the outer membrane. This outer membrane distinguishes our gram-positive from our gram-negative. Now, here's the interesting thing. The medications that we see as cell wall synthesis inhibitors target peptidoglycan. They may target it a different way, but since peptidoglycan is an integral part of bacterial cell wall, targeting its synthesis is how our drugs work, our cell wall synthesis inhibitors. That's why we call them cell wall synthesis inhibitors. So now how do they interfere with the cell wall synthesis of of bacteria? We can also say, how do they interfere with peptidoglycan formation, since peptidoglycan serves as an important component of the cell wall. In fact, in gram-positive, it makes up the whole cell wall. So we will start with the first group, the beta-lactam antibiotics. So cell wall synthesis inhibitors can be grouped into two, the beta-lactam antibiotics and the glycopeptides. The beta-lactam antibiotics covers 4 different antibiotic classes: penicillins, cephalosporins, carbapenems, and monobacter. All these 4 are called beta-lactam antibiotics. They are called beta-lactam antibiotics because in their chemical structure, they all have this beta-lactam ring. So let me show that to you in a picture form. So you note that this is penicillin and this is the beta-lactam ring. This is cephalosporins, and you can see the beta-lactam ring in yellow. Carbapenems, you can see the beta-lactam ring in yellow. And monobacters, you can see the beta-lactam ring in yellow. The point is, all these antibiotics have the beta-lactam the beta-lactam ring, and that's why we call them beta-lactam antibiotics. Know that the presence of the beta-lactam ring is what confers on them the ability to inhibit cell wall synthesis. And we will look at how they do that shortly. We should also know that all the beta-lactam antibiotics also contain a carboxyl or sulfonyl group, a group that confers on them water solubility, and therefore elimination occurs through the kidney. renal excretion. Because of the carboxyl or sulfonyl group, these molecules are water soluble, and being water soluble and polar in nature, are eliminated or removed by the kidney or the renal system. Now, because they all share the beta-lactam ring and the carboxyl group, they all seem to also share similar pharmacokinetic properties and similar mechanism of action. So this means if an individual responds well to penicillins, it is likely they will not get a reaction, or they will respond the same way they are responding to their penicillins or cephalosporins or carbapenems or monobactam. So we say that there is also cross resistance. If there's resistance here, you may get resistance. The organism may also resist cephalosporins, carbapenems, even sometimes monobactams. But it's been shown that for monobactams, this seems to have a more robust activity against bacteria because of the single beta-lactam ring compared to the others. All right. So this is so important that we understand and we know that all beta-lactam antibiotics have a beta-lactam ring, a carboxyl group that makes them water-soluble and hence eliminatable by friends and foe. All right. Now, we said that the beta-lactam ring gives these antibiotics the ability to interfere with cell wall synthesis. So how do they do it? There are various proteins or enzymes which bacteria uses to create the cell wall. These enzymes are known as transpeptides. Transpeptides. And because the activity that the enzymes go through when bacteria is forming cell wall is named after them, we will say transpeptidation reaction. The reaction caused by the transpeptidase enzyme, or reaction caused by penicillin binding proteins. So the point is this: the beta-lactam ring binds to this enzyme, and since the enzyme is an integral part in the cross-linking of the peptidoglycan layer, by inhibiting the enzyme and its activity, we are able to stop the transpeptidation reaction or the cross-linking of the peptidoglycan layer. Once that is inhibited and stopped, bacterial cell wall that is formed is weak, and that results in the death of the bacteria. So mostly beta-lactam antibiotics act as bactericidal agents. All right. Please, I hope that makes sense. Let me just take it again for the sake of you, those who didn't get it, or those who just need a repeat to cement it in their minds. What I'm saying is the presence of a beta-lactam ring in the beta-lactam antibiotics confer on them the ability to kill some bacteria. And how do they do that? Bacteria has an enzyme, transpeptidase, which is in the bacteria cell. It's also sometimes called penicillin binding proteins. So PBP, penicillin binding proteins. Now, these enzymes are involved in a reaction that is called the cross-linking of the layer. If you still remember when we talked about the characteristic differences between gram-positive, gram-negative, one of the things that is there is that gram-negative have single layers, whereas gram-positive have multi-layers. Now, each layer is made up of the peptidoglycan. So to create that layer, there must be a cross-linking of proteins. Now, that is done by the enzyme transpeptidase. Therefore, that cross-linking process is called transpeptidation reaction. So basically what we are saying is when you give a beta-lactam antibiotics, it binds to the transpeptidase enzyme, thereby interfering or preventing the transpeptidation reaction, which is the cross-linking of the peptidoglycan layer. This results in weak cell walls that easily lyse and breaks, pouring content of the bacteria out, resulting in the death of the bacteria. All right. So that is the way the beta-lactam antibiotics work. That's the way they work. Second way by which cell wall synthesis is inhibited. You see, the first way we said through the beta-lactam beta-lactam ring. Now, the second way is by a mechanism which normally works in gram-positive organisms. This mechanism that we're going to talk about doesn't work in gram-negative organisms. And this is the mechanism by which the class of antibiotics known as the glycopeptides work. The glycopeptides. And how do they work? In gram-positive organisms. Basically, these has to be able to get to the D-alanine terminus of the peptidoglycan. There is a peptidoglycan precursor when the linkage is occurring. Now, at the end, at one terminus, at one end of the peptidoglycan precursor, there is the D-alanine, and D-alanine is basically telling you about the protein, the amino acid involved there. Now, this D-alanine end must be binded in the transglycosylation reaction, which is a very necessary reaction in cross-linking, or what we just finished calling the transpeptidation reaction, right? So for transpeptidation reaction to occur, we will have to have the linkage made, a cross linkage. Now, before that cross-linkage is done, there must be the D-alanine terminus joining, the binding. This is called the transglycosylation reaction, which is a reaction controlled by enzyme activity. So basically, when we use glycopeptides directly, they bind to the D-alanine terminus and interferes or stops the transpeptidation reaction through stopping the transglycosylation reaction. Basically, what we are saying is that glycopeptides also inhibit cell wall synthesis. Howbeit, they do not use the beta-lactam ring, rather, they inhibit the D-alanine terminus. Basically, they just bind to that site of the peptidoglycan and prevent the cross-linking. So in both cases, we are stopping the transpeptidation reaction using two different methods. Beta-lactam ring binds to the enzyme that is going to do that, whereas the glycopeptides bind directly, not to the enzyme, but directly to the D-alanine terminus, to the peptidoglycan directly, and prevent the transpeptidation reaction. So you note that both of them inhibit or interferes with cell wall synthesis. That's why we call them cell wall synthesis inhibitors. So when we say cell wall synthesis inhibitors, we have two groups: the beta-lactam antibiotics that work by binding to the enzyme through the beta-lactam ring, and then we have the glycopeptides that work directly by binding to the D-alanine terminus of your peptidoglycan. Both activities or both class of antibiotics ultimately inhibit cell wall synthesis, ultimately inhibit bacterial cell wall synthesis. And that is why, or that is how they work. So their mechanism of action. I hope that makes sense to you as well. All right. So once we have disrupted this activity, basically they result in the cell's death. Now, basically we can again see that because these class of drugs bind to the D-alanine, the D-alanine terminus is not available for the enzyme required for the transglycosylation reaction. And that is how come it leads to the death of the bacteria. All right. So basically, there is a disruption in the cell wall construction. Weak cell wall, which will lyse, which will break apart and cause the death of the bacteria. So this is the first method, first mechanism, cell wall synthesis inhibition. Note there are two classes of antibiotics that work like this. The beta-lactam antibiotics, which work through the beta-lactam ring, and the glycopeptides, which work by directly binding to the D-alanine terminus. Now, if you get that concept, then you can see that when we talk about penicillin, cephalosporins, carbapenems, or monobactams, we automatically know these are cell wall synthesis inhibitors. And then when we talk about glycopeptides, we also know These are cell wall synthesis inhibitors. The only distinction, important distinction, is that glycopeptides are active or effective only in gram-positive organisms. So never use them for gram-negative organisms. Thank you so very much. We will continue our lesson in the next video. Have a wonderful rest of the period. Please don't forget to subscribe for more. You are blessed. You are blessed. Amen.